And so, in conclusion, glycans play an essential role in health and disease. Are there any questions? Yes, what's just glycan? An infection here in the alveoli of the lungs can be deadly, and lymphocytes patrol the area diligently. When a pathogen is detected, neutrophils release elastase, an enzyme that degrades bacterial cell walls and disrupts biofilms. It also breaks down the extracellular matrix to help immune cells extravasate into the tissue. If that part makes you a little nervous, it should. Neutrophil elastase has to be quickly neutralized before it can damage the lungs.
It went active the moment it was launched. It's acquired us and is homing.
Fortunately, the liver secretes Alpha-1 antitrypsin, a serine protease that degrades elastase.
Way to go, Dallas!
However, there's a genetic disease called Alpha-1 Antitrypsin Deficiency, in which a mutation in the SERPINA1 gene prevents the liver from secreting Alpha-1 antitrypsin.
That torpedo's still active. It's searching for another target now.
Without it, neutrophil elastase hydrolyzes the elastin and collagen in the connective tissue of the lungs, leading to emphysema and chronic obstructive pulmonary disease. Alpha-1 antitrypsin deficiency is partly caused by an error in glycosylation. Now, glycosylation is an enzymatic process in which a glycan is attached to a protein or a lipid. What's a glycan, and what does it have to do with alpha-1 antitrypsin deficiency? Glycans are carbohydrates, which are organic compounds containing carbon, oxygen, and hydrogen. But one of the tricky aspects of organic chemistry is the concept of symmetry. When carbon attaches to 4 different groups, the orientation of each group suddenly starts to matter. The mirror images of the chiral carbon are classified as either L or D in enantiomers. Thanks to these chiral carbons, there's an enormous number of different sugar molecules that could exist, but fortunately the D form of sugars is much more common in nature. And surprisingly, humans only use about 10 different monosaccharides to make the full range of glycans. Glucose is the most important sugar in the body and can be converted to or from other monosaccharides as necessary. But glucose is usually found in ring form, and this introduces yet another chiral carbon. So strictly speaking, this glucose molecule is called alpha-D-glucopyranose. D specifies the enantiomer at carbon 2, pyranose refers to the ring form, and alpha refers to the position of the hydroxyl group on this new chiral carbon. Glucose is the energy currency of the body, and the levels rise and fall throughout the day. Insulin resistance and diabetes mellitus can lead to dangerously high levels of glucose in the blood. Hyperglycemia can cause organ damage, and hyperosmolar hyperglycemic state, which can lead to coma. Aside from directly measuring the blood glucose levels, we can also look at HbA1c as a way to measure long-term trends in blood glucose levels. HbA1c is formed because a small amount of glucose spontaneously binds to hemoglobin. In this case, we say that the hemoglobin is glycated because there's no enzymes involved. But more commonly, enzymes called glycosyltransferases attach sugar moieties to proteins in the process of glycosylation. One example of this is the human ABO blood group, which differ from each other by only one sugar. All 3 share the same core, but in people with type A blood, the enzyme N-acetylgalactosaminyltransferase attaches an N-acetylgalactosamine to the core. Conversely, people with type B blood have a galactosyltransferase that transfers galactose instead, and people with type O blood completely lack the enzyme for this, so the core remains as it is. And glycans are ubiquitous. Vascular endothelial cells are covered by a fuzzy coating of glycolipids and glycoproteins called the glycocalyx, which forms a protective barrier. Many glycans also play important roles in immunity, including recruitment and adhesion of lymphocytes. While glucose is the most abundant sugar in the blood, other sugars such as fructose and galactose pass through the portal vein on the way to the liver. Larger sugars are broken down in the small intestine, and some ingested carbohydrates can be enormous. A single molecule of amylopectin can contain millions of glucose residues. Salivary amylase begins breaking down starch in the mouth, and enzymes in the small intestine break carbohydrates down into individual monosaccharides that are processed in the liver. We tend to think of the cell surface as a featureless desert, but the plasma membrane is more like a dense forest, covered with all manner of receptors, transporters, adhesion molecules, and glycosaminoglycans. More than half of all human proteins are glycosylated, but that number jumps to 90% among cell surface proteins. These post-translational modifications perform a variety of functions, including cell-cell recognition, cell signaling, and immune modulation. Most glycoproteins are either O-linked or N-linked. In O-linked glycoproteins, such as the ABO blood group, a glycan chain is attached to an oxygen atom in either a serine or a threonine residue. Glycosaminoglycans, such as heparin sulfate, chondroitin sulfate, and hyaluronic acid, are key components of the extracellular matrix and consist of long chains of repeating disaccharide units. The other major category is N-linked glycoproteins, where a glycan chain is attached to a nitrogen atom and an asparagine residue. N-linked glycoproteins can be extremely diverse, ranging all the way from antibodies to the insulin receptor, but they all share a common core consisting of N-acetylglucosamine, often called GlcNAc, and mannose residues. N-linked glycosylation plays an important role in protein folding and stability. This is where glycosylation comes in Alpha-1 antitrypsin deficiency. The disease is caused in part by defective N-linked glycosylation in the liver. The liver plays a critical role in carbohydrate metabolism and energy balance for the body. One of the first things you might notice about hepatocytes is the large number of glycogen granules. The pancreas regulates the ability of the body to utilize glucose, but it's the liver that's responsible for making sure there's enough glucose to meet the body's energy demands. Fats can be broken down to provide energy as needed, but that takes time, and the liver needs a large amount of glucose on hand to respond quickly to changing energy needs. Unfortunately, the liver cannot just store a large amount of free glucose in the cytoplasm. That would severely disrupt the osmotic balance and cause cells to burst. Instead, the liver stores glucose in the form of highly branched glycogen molecules. Glucose residues can be cleaved from multiple branches at the same time and quickly released into the circulation. But the liver exports many other substances, including N-linked glycoproteins like Alpha-1 antitrypsin. The sequence and branching patterns of N-linked glycoproteins are highly specific, and this poses an interesting question. We know that the amino acid sequence of a protein is unambiguously coded by the sequence of nucleotides in the DNA, but how can the genome encode the branch structure of a glycan? It helps to think of it as an assembly line. The machines on the line and the order they're arranged in determine what can be produced. The expression of different types of glucosyltransferases in a cell determine which machines are on the assembly line. But how can the order be controlled? Well, first let's start with the protein backbone. The SERPINA1 gene is transcribed and exported from the nucleus, and the messenger RNA binds to a ribosome. The ribosome attaches to the ER membrane and feeds the translated protein into the lumen. At the same time, the glycan core is assembled separately on a molecule of dolichal phosphate embedded in the ER membrane. Importantly, though, this occurs on the cytosolic side of the ER. First, the enzyme GlcNAc1 phosphotransferase transfers a molecule of N-acetylglucosamine, or GlcNAc, to the dolichal phosphate. Then another GlcNAc is added to the first. In the next step, a series of 3 different mannose transferases attach 5 mannose residues to the growing core. At this point, an enzyme appropriately named flippase flips the entire glycan structure into the lumen of the ER, and another set of mannose transferases attaches 4 more mannose residues for a total of 9. Finally, glucosyltransferases attach a set of 3 glucose molecules to the structure. This complex process is coordinated both through strong substrate specificity as well as by the physical separation on either side of the ER membrane. An oligosaccharyl transferase transfers the completed 14-sugar core to an asparagine residue on the Alpha-1 antitrypsin polypeptide. Oligosaccharyl transferase recognizes a specific 3-amino acid motif, starting with asparagine and ending with serine or threonine. Alpha-1 antitrypsin has 3 of these motifs, and glycans are added at asparagines 46, 83, and 247. But what purpose do these glycan chains serve? Let's go back to those 3 glucose molecules for a minute. Well, this core structure is altered quite a bit before release. The 3 glucose molecules are never included in the final structure. Why not? Because they're used exclusively for quality control. ER glucosidases 1 and 2 remove the first 2 glucose residues, which causes the chaperone proteins connexin and calreticulin to bind to the nascent protein. These chaperones help make sure the protein folds correctly. If there's a problem, the chaperones disengage, and another enzyme, UDP glucose glycoprotein glucosyltransferase, or just UGGT, reattaches a glucose molecule. This is a signal for the chaperones to try again. But if the protein repeatedly fails to pass this quality control check, the glucose molecules are never removed. The presence of these glucose residues blocks the protein from leaving the ER and triggers its destruction through the ER-associated degradation pathway. On the other hand, if the protein is folded correctly, ER glucosidase 2 removes the final glucose, allowing the protein to be loaded into COP1-coated vesicles. The vesicle is transported by motor proteins to the Golgi apparatus, where it docks and releases the immature protein into the cis-Golgi network. Here we see another way that the order of assembly can be enforced, by spatially separating the processing steps between the cis, medial, and trans Golgi compartments. In the cis Golgi compartment, any remaining glucose residues are removed, then mannosides 1 and 2 remove 4 of the mannose residues. Next, N-acetylglucosaminotransferase 1 attaches a GLUCNAC residue, and the maturing protein is transported to the next compartment. In the medial Golgi compartment, mannosides 2 trims 2 more mannose residues, and N-acetylglucosaminotransferase 2 adds a second GLUCNAC residue. Then galactosyltransferase adds galactose residues onto the terminal GLUCNAC residues, and the protein moves to the trans Golgi compartment. Sialyltransferases attach sialic acid onto the galactose residues, which helps to stabilize the structure and improve its half-life in circulation. Finally, fucosyltransferase adds a fucose residue to the core GLUCNAC. Fucosylation plays an important role in cellular adhesion and modulation of the immune response, and is often elevated in cancer. The mature glycoprotein is sorted and packaged into secretory vesicles, which bud off from the Golgi and merge with the cell membrane, releasing alpha-1 antitrypsin into the circulation, where it eventually reaches the lungs. Or at least that's how it's supposed to work. But in patients with alpha-1 antitrypsin deficiency, alpha-1 antitrypsin may not even reach the Golgi apparatus, let alone the lungs. SERPINA1 mutations, such as the Z allele, in which glutamic acid at position 342 is replaced with lysine, change the 3D structure of the protein, leading to incomplete glycosylation. The misfolded protein fails the quality control step, and the glucose markers are not removed, preventing the protein from leaving the ER. Defective alpha-1 antitrypsin begins to accumulate in the ER. Worse yet, the misfolded proteins tend to aggregate and form large insoluble polymers, leading to ER stress and inflammation. The liver is a remarkably resilient organ, but chronic ER stress damages the liver and can eventually progress to cirrhosis or even hepatocellular carcinoma. And then what's the treatment for alpha-1 antitrypsin deficiency? The main treatment is augmentation therapy. In this treatment, functional alpha-1 antitrypsin derived from plasma is administered intravenously through weekly infusions to help protect the lungs against further damage. Now, this doesn't help to repair existing damage to the lungs, but beta-adrenergic agents and ipratropium bromide bronchodilators can help to maximize the remaining lung function. This helps the lungs, but alpha-1 antitrypsin deficiency also damages the liver, and in severe cases, transplantation of either the lungs or the liver may be needed. Other therapies in various stages of development include synthetic chaperone proteins and gene therapy or gene editing to restore functional copies of the SERPINA1 gene. We tend to focus on the role of carbohydrates as an energy source, but glycans perform many additional functions essential for immunology, protein folding, cell-cell communication, and structural integrity. And so, in summary, glycans play an essential role in health and disease. Are there any questions? What's a glycan?